I'm Jenny Flynn, and I'm going to be doing my dark reactions explanation, and the dark reactions are the Kelvin reactions. So there's light and dark reactions in the cell, and the light reactions must take place in light because they require the light photons, but the dark reactions only require what's already in the cell or what the product of the light reaction is. So they don't necessarily have to take place in light, but they do require light to hit the cell before it can occur, at one point at least. And my diagram is a little messy again. And before I start though, I want to clarify something that I didn't mention in my first video, and I just don't want to miss it. The chlorophyll center, and I already uploaded my video, otherwise I would change it. But the chlorophyll right here that I mentioned is just going to be the reaction center of chlorophyll A, because that's chlorophyll A is what we're using in that reaction. And so that's that. And then I don't think there was anything else I needed to clarify. If I think of anything or remember anything while I'm explaining this, I'll go through it. But I'm going to try and go through this a little bit quicker than I went through the last one because I tend to ramble. So this is going to start with carbon dioxide, which is already going to be present in the cell. Carbon dioxide, which is already going to be present in the cell. And this 5-carbon chain right here, or 5-carbon molecule right here, which is going to be called the ribulose biphosphate. And all that is just 5 carbons attached to each other. They probably have hydrogens attached, but for this reaction, the hydrogens aren't very necessary to mention. So they're going to combine right here, and they're going to combine. Carbon dioxide is going to be broken down by using an enzyme, RUBP carboxylase, which is just kind of a another term it's used to when I was looking it up online is Rubisco. So, but for the terms of this paper, we'll say RUBP carboxylase. So that's going to help carbon dioxide separate the carbon molecule from these oxygen molecules right here. It's going to kind of help break the bond. That way this carbon can be used here, because you're going to see we need 6 total carbons. So we need the carbon from this and the 5 carbons here from the ribulose biphosphate to make this these 2 molecules here, which are going to be 2 molecules of 3-phosphate glycerate. And that's going to be the product here. So these 2 molecules are going to go through some reactions which occur right here. You're going to use the ATP from the light reactions, and it's going to go through several reactions, and it's going to also use the NADPH from the light reactions here. So what's going to happen is these are both going to be oxidized, so they're going to lose electrons. So right here you're going to lose this phosphate, and this phosphate is going to help in the reaction of these glycerates. And then this ADP, once the ATP loses the phosphate, this ADP is going to go back and it's going to be used in the light reactions. The NADPH is going to lose a hydrogen, and it's going to just be oxidized back to NADP+, and that NADP+ is going to go back and be used again in the light reactions as well. So during all of these reactions that are taking place right here, a new molecule is going to be formed, and that molecule is going to be called 3-phosphate glyceraldehyde. And the 3-phosphate glyceraldehyde from there can be broken down into built up into the glucose molecule, which is right here. So the glucose molecule is kind of just the product that you want to end all of the dark reactions with. It's the goal of the dark reactions. And I just remembered something from the light reactions that I wanted to clarify. The extra molecules that are left over, not from the glucose. Now this has to take place several times to form that glucose molecule, because each time you're only going to be getting a certain amount of product, and it's not going to be enough product to make that glucose. So this has to occur multiple times, several times, and any leftover is going to be kind of left out, and this carbon dioxide right here It's going to be one of the the extras that you don't need for the glucose to form. You're going to use that up here. That's what you used in the original starting molecule. Now, the part that I just mentioned before isn't super relevant, but it is does play a certain role in this. So the plastoquinone that I mentioned in the first video, that is the enzyme that attaches to the hydrogen, when that loses the hydrogen, it's going to change into something called a plastoquinol. Not sure about the pronunciation on that. That's basically the ol is like making an alcohol group. So, but that is going to be used. That's going to be one of the products of the light reactions that's going to help in this reaction during the hydrogens breaking in this reaction, and it's going to help this carbon dioxide here just as well as the RUBP carboxylase. I think I covered everything. I'm just reviewing my words real quick to make sure I covered and hit over everything. So I think I hit all the main points. I can't think of anything that I left out. I hope I didn't leave anything out. Nothing important, at least. One thing, I guess, yes, I'm glad I waited. The cytochrome B6F complex. I knew there was something that's missing. That is more of a location. That's more of a location. And I don't have a good spot to put it in here because of the way I have this drawn. But let me see if I can use one of these other pictures that I pulled off the interwebs. Don't see one. Let me see. It actually occurs in the light reactions. I should have mentioned it in the first video. It's the when these hydrogens, where did I draw it at? These hydrogens that are split right here, right here. So the oxygen, the water molecule. I'm sorry. It's gonna be, scratch what I just said. It's gonna be right here where these electrons are being pulled apart, like they're being dispelled from the chlorophyll before they're used in these ADP and the broken water molecule with the 2 hydrogen. That right there is going to be, drew brackets around it. That right there is going to be the complex. So these electrons can actually be thought of as electrically charged hydrogens. So, and that would be the cytochrome B6F complex, which I'm glad I remembered to mention. So that is the end of my reactions video. So I'm sorry that I added some of the light reactions to this one. I just, whenever I was making my dark reaction schematic sheet here, I didn't mention the some of the words, and I went to go look them up again, and they were for the light reaction, so I just want to throw them in there. So I'm sorry about that, but that's it. So I hope you have a great rest of your weekend.